So I'm going to move away from the debate about whether place cells exist and whether there's a map, which by the time I came on the field, at least some of us were convinced. I don't think that everybody was yet convinced. But I want to address the question about how they get to be place cells, which is something that has a long and checkered history and is still a matter of intense debate. And this is going to, of necessity, be a rather brief tachistoscopic sketch. So in the beginning, there was O'Keefe and Dostrovsky, as John's already mentioned, and I just want to point out what I read as the main conclusion of that paper was that the activity of cells in such a map would specify the direction in which the rat was pointing relative to environmental landmarks and the occurrence of particular tactile, visual, etc. stimuli while facing in that direction. So in a sense, although they were spatial cells, according to that view, they were being driven by some high-level integration of sensory features. And around 1982 or 81, when Carol and I joined John's lab, John had already set up a video camera and was starting to try to move towards quantitative evaluation. And I had a little computer programming skills, so I sat down and programmed the computer to do quantitative analysis of firing rates on the 8-arm maze. And what we found, which you can see in this ancient picture here, was that these cells almost uniformly fired exclusively in one direction on the 8-arm maze, or the amount of variance that was explained by firing in the other direction was no more than chance. And I wrote, the pronounced directional selectivity of complex spike cells suggests that they fire in response to complex but specific stimulus features in the extramaze world rather than to absolute place in a non-egocentric space. John O'Keefe did not like that. But he put his name on the paper anyway. And I think that sort of characterizes our relationship from then on. So I had the view, or I had the idea from this, that place cells were what could be called local view cells, with view encompassing all sensory modalities, that they were just high-level sensory cells. But there was one factor in our study that sort of didn't fit into that picture, and that was the fact that place cells also had a velocity characteristic. So they were very highly tuned, as all cells in the hippocampus are, to running speed, and I wasn't sure what that meant at the time. Well, a few years later, these upstarts in New York came along also with a quantitative tracking system, but they didn't use an 8-arm maze. They used this famous cylinder with a cue card. And what they concluded was that in the present work, we observed that orientation was at most a second-order factor for place cell firing. And in a subsequent paper, they said, there is no intrinsic directional specificity of place cell firing in the cylinder. And this is what gave me pause and made me start to rethink what might be going on in the system. But what really changed my view was when Jim Ranck came along in 1984 at the neuroscience meeting. He had a video system there and showed us movies of head direction cells in somewhere in the presubiculum, dorsal presubiculum. And so not only was this sort of evidence that there was a map, but there was also a compass. And just for historical sake, I think it's worth pointing out that this was the point, I think, when the neuroscience community cottoned on to the idea that it was actually a map. Because you know, you could say it's a map or not a map from place cells, but when you saw a compass cell, you had to say, oh, a map and a compass in the same place is just too much of a coincidence. And that was when the field really started to take off. And I think that the referees of the Nobel Prize overlooked something very important when they didn't invite Jim Ranck to share in that prize, because I think that this was the observation that really set the field going. Not to denigrate the important contributions, obviously, of the other recipients. So I began playing around with head direction cells actually before the famous Taube et al. Paper came out. And one of the things we observed in the lab was that, A, they fired in any given place you wanted to put them. B, they fired in the darkness. And C, if you slowly rotated the box the animal was in, they changed their preferred angles to maintain consistency with the box. And that was when I said, my God, I've read about this. I think Darwin started it, and there are a few other people. Path integration. This is a path integration system. And so we did some computational modeling, which I won't go into in detail, but it was a sort of continuous attractor type of model, not the early version, but eventually we came down to that. I should mention that Ketchen Zhang published a paper more or less the same time with more or less the same idea. A key prediction was the existence in a kind of hidden layer of cells that were conjunctive for the direction the animal was facing and the speed at which he was, or the angular velocity at which he was turning. And those cells were subsequently confirmed in a number of parts of the brain. So if the head direction system could be a path integration system, why not the place cell map itself? And there were a lot of data, and a lot of data has accumulated over the years, only some of which is illustrated here. First of all, the fact that they continue to fire the same place in the darkness. Secondly, Muller et al.'s demonstration that in the cylinder, they don't care which direction the animal's facing, so it can't be local view cells. In my student Kati Gothard's experiments, she showed that the cells fired in relation to the distance the animal had moved from a fixed reference point, even though the fixed reference point was moving around in the laboratory, so it couldn't be the external cues that were driving the cell. When Jim Knierim was working in my lab, he showed that head direction cells and place cells form a tightly coupled network, that when you get drift error in one of the systems, the other system gets exactly the same amount of drift error. And then also, if you run an animal in an environment explicitly designed to test the idea about whether the scale of the spatial representation is dictated by local features of the environment, we found that with animals running back and forth on this kind of a track, there was virtually no effect on the size of the place field, in spite of the fact that one side of the maze had a lot of rapid changes in the sensory features. The scale stayed constant. And about the same time, Min Zheng in my lab showed that as you moved successively more ventral in the hippocampus, the scale got bigger and bigger. So that makes it very hard to believe that these cells are configured, at least initially, by the sensory features of the environment. There's something intrinsic going on. And so what we and also Ketchen Zhang did was to extend the one-dimensional attractor model for head direction cells to a two-dimensional attractor model in which we could take the head direction system and project it through a hidden layer of cells that are conjunctive for position and head direction and had movement sensitivity and create a model for path integration. One of the important features of this model was that the role of the external cues was not to set up the firing or to change the firing, but simply to realign the whole system when it got out of whack. Okay, so to correct for drift error. But in our original thinking, the idea was that this top-down influence would correct the path integrator, but wouldn't itself affect the firing of the place cells. The central dogma at the time was that place cells didn't change their firing rates much when you changed what the animal was doing or changed features. But that view crumbled, and there were many papers which led to the crumbling, but eventually the field emerged with this concept of rate remapping, which is simply to say that although place fields are not set up and determined by the external cues, the external cues control the firing rate of the cell in its place field. So the external cues don't determine where the cell fires, but they determine how much it's going to fire in a given place. And following from that concept, we went back to address the apparent conflict between the Kubi-Muller-Ranck lack of directionality with the directionality we saw so clearly on linear tracks. And lo and behold, when we looked at the very first lap on a linear track that the animal experienced, the cells were totally non-directional, just as they are in the open field. But as the animal runs even 1 or 2, 3 laps, very quickly, a strong directional dependence emerges. So it was clear that the external cues were powerful modulator of the firing, but not determining necessarily where the cells were going to fire in the first place. So coming back to this sort of theoretical model, one of the things that it predicted, just like the conjunctive cells in the head direction model, this two-dimensional model required conjunctive cells that were conjunctive for place and head direction. And we, of course, didn't see them in the hippocampus, nor did we see in the hippocampus, I should just mention as a side, when Alexei Samsonovich simulated this, he did it on a torus, so the place fields repeated regularly in a regular lattice. We never saw that. We looked for it in the hippocampus, and we didn't see conjunctive cells, but sure enough, conjunctive cells were later found in the entorhinal cortex, which is, the fulfillment of a prediction of this path integration-based model for the spatial map. Well, I can skip this, except that the key prediction here was the conjunction of position times head direction cells, which I just showed you, plus this movement sensitivity. So the key to this model is that what moves the hill of activity around the torus is nothing to do with the external world at all, but simply to do with the animal's sense of motion, how fast he's running. So we looked in the hippocampus, and we found that sure enough, as you move from the dorsal hippocampus to the more ventral hippocampus, the scale gets bigger, and at the same time, the strength of the speed signal diminishes. So in the dorsal hippocampus, there's a very sharp relationship between the theta amplitude and running speed. As you go to the middle hippocampus, it gets more flat, and as Yuri Basaki has shown, as you get to the very ventral hippocampus, there's virtually no or detectable change in the amplitude. The intrinsic oscillation frequency of place cells varies systematically from dorsal to ventral, and the firing rates also vary systematically from dorsal to ventral. And indeed, if you now remove some of the self-motion cues by having the animal, for example, get up and drive instead of walk, the place fields suddenly get bigger. They get small again as soon as he gets back on to walking around. So that really, I think, summarizes a lot of the evidence for the notion that place cells and the hippocampal system in general is being strongly driven by some form of path integration mechanism. Where am I going here? But there's a problem with this model, And the problem is that if you have a continuous system, it's not possible for the hill of activity to simultaneously move at different speeds around the torus. So a hard prediction of the model was that actually the path integration system must be modular. We called it grid scale or spatial scale quantization. We said that there had to be a number of modules in the system, each with a different gain of the self-motion signal in order to generate different scales along the septotemporal axis. And sure enough, the Mosers obliged us by showing that indeed there is grid scale quantization, and it occurs in a very interesting geometric pattern where there's a factor of 1.5 or 1.7. Okay, anyway, there's a constant factor, and I'll come back to the importance of that in a minute. So now we want to talk about how place cells get there in the first place. Well, when people saw grid cells with different spatial scales, anybody who had even an inkling about Fourier synthesis said, oh, okay, if you add together place fields or grids of different sizes, you're going to get a unique bump. At least locally. It may continue and repeat at great distances, but you can get with a little thresholding. And so a couple of papers described this, which was the standard model in people's thinking for how place cells emerged in the hippocampus downstream from the medial entorhinal cortex path integrator. So we set out to test this. Jake Orman developed a nice, difficult setup where he could put cannulae in at various depths along the dorsal to ventral entorhinal cortex. Of course, the Fourier synthesis model predicts that if you knock out the high frequencies, the place fields will get bigger. If you knock out the low frequencies, the place fields will get smaller. And so he did that very difficult experiment, 3 years of his postdoc, maybe 4, doing that one experiment, but the results finally came out and they weren't all that clear. No matter where we put lidocaine in the entorhinal cortex, the place fields got bigger in the hippocampus. Okay? But there was a systematic difference, and the systematic difference was that the more ventral you got, the less they got bigger, until there was almost no change. Okay, so injections in the dorsal entorhinal cortex really did make the fields bigger, but injections successively more ventral had much less effect. And so we thought, well, this is sort of consistent if you make a few other assumptions about things about knocking out the self-motion speed may have contributed to the fact that everything got bigger. And then along came my good friends, John Kirby and Steve Fox, and they published a nice commentary in which they showed, well, hey, we actually simulated this, and if you assume that there's this geometric expansion of the grid scale and not a continuous one, then knocking out the slow frequencies didn't have much of an effect. So we like that, but I'm not entirely convinced anymore that that's the answer. One of the problems is that all of these theories and many of people's thinking about the role of the hippocampus as a memory system and so on assume that all cells are created equal, and that in order to get orthogonalization of representations and to get maximum information content, every cell in the hippocampus has to have an equal chance at having a place field. And it turns out that that's the furthest thing from the truth. And that's why I say seahorses have long tails, because it's been known actually for many years that hippocampal cells have a highly skewed firing rate distribution, that there are many cells with very, very low firing rates, almost never firing at all, and a few cells with high firing rate, and that seems to be consistent across different subfields. And then we moved to looking with immediate early genes as an attempt to capture all the cells in the hippocampus and see how they distribute. And we tested this sort of uniform random draw with replacement model using immediate early genes by running animals in one environment or successively more environments. And if you knew the probability of activation in one environment, you could predict what the probability of activation in n environments would be based on uniform random draw with replacement. And it turns out when you do this on many, many experiments, they all undershoot the prediction greatly. The conclusion being that many hippocampal cells have a very low probability of being allocated, no matter where you put the animal. Well, looking back to the work of Urey and his colleagues, we should have known this because they did these elegant experiments with simultaneous intracellular recording and extracellular tetrode recording in slices. And what they concluded was that if you looked at how far away the tetrode could be from a cell and pick up the spike, and you looked at the number of cells it should see, you should see, 100 or more cells on a given tetrode at a given time, which is never the case. You're lucky on good days you see 5 or 6, most of the time 2 or 3, sometimes never. And so that leads to the idea that almost 90 or 95% of CA1 neurons are virtually silent and cannot be allocated to the spatial map no matter what you do, at least on a given day. We don't know yet whether this is plastic. We also know that if you look at the number of fields that a given cell forms, it's not a Poisson distribution, as you would predict from random draw. And then there was this beautiful study by Rich et al. In 2014, which really quantified this very nicely on a very long 50-meter track. And what they showed was that, indeed, the allocation of fields to a given cell is a random Poisson process. It's unpredictable, it's memoryless. But if you look at the population of cells, there's a huge variation in the number of fields or the probability that the cell will have a given number of fields. And so that again shows this highly skewed long-tailed distribution of place fields. So it looks like, contrary to the sort of Fourier model, that allocation of cells or place fields to a cell, at least for a given cell, is a purely random process. It's indistinguishable from random, which is not what you'd predict if it was a Fourier synthesis model. And so I'm left now thinking about what probably is going on. And my thought is that has something to do with the relative weights of the medial and lateral perforant paths, medial entorhinal, lateral entorhinal input. That when a cell is activated in an environment by some random selection process that we don't know, the first thing that hits it and that it really experiences is the medial entorhinal input. And this thinking goes back to my early days looking at medial and lateral perforant path, where we found that the terminals of the medial perforant path have a high transmitter release probability at rest, whereas the terminals of the lateral perforant path have a very low release probability at rest. Can be potentiated, but start off weak. And so what I'm thinking now is that there's some intrinsic difference. It's not that the lateral perforant path and sensory inputs can't generate place fields. They probably can. They can certainly modify over time the firing rates of these cells, but what initiates the firing is the output of the path integrator system in most situations. And that's it. Thank you.